Welcome back to MedMind.
This quiz contains MCQs from the first chapter, the cell as a unit of health and disease, and the topics covered are mentioned below. So before we start, hit the like button and subscribe our channel. Let's get started. What does the term pathology literally means? A. Study of disease. B. Study of life. C. Study of cells. D. Study of suffering. Answer is D. Study of suffering. Who suggested that disease originates at the cellular level? Watson, Crick, Virtue, or Pasteur? Correct answer is C. Virtue. The human genome was sequenced at the beginning of which century? A. 19th, B. 20th, C. 21st century, or D. 22nd century? Correct answer is C. 21st century. Approximately how many protein encoding genes are there in the human genome? 10,000, 20,000, 30,000, or 40,000? B. 20,000. Protein encoding genes constitute about what percentage of the human genome? 1.5%, 10%, 20%, or 50%? Correct answer is A. 1.5%. The majority of the human genome is devoted to building proteins, B. Regulating gene expression, C. Disease immunity, D. Organ development. B. Regulating gene expression. Non-coding regions of the human genome are sometimes referred to as A. Exons, B. Codons, C. Dark matter, or D. Genetic mutations. C. Genome dark matter. What percentage of the human genome is transcribed? 50%, 85%, 95%, or 100%? 85%. Which organisms have about the same number of protein encoding genes as humans? A. Worms, B. Monkeys, C. Elephants, D. Frogs? Correct answer is A. Worms. How much of the human genome is involved in the gene regulation? 40%, 60%, 80%, 90%? Correct answer is C. 80%. The human genome consists of approximately a billion DNA base pairs, 2.5 billion, 3.2 billion, or 5 billion DNA base pairs. Correct answer is C. 3.2 billion DNA base pairs. What are the two most common forms of DNA variation in the human genome? SNPs and inversion, CNVs and mutations, SNPs and CNVs, exons and introns. C. SNPs and CNVs. Single nucleotide polymorphism, that is SNPs, are usually monoallelic, triallelic, biallelic, or quadallelic. Correct answer is C. Biallelic. Approximately how many SNPs have been identified? 1 million, 3 million, 5 million, or 6 million? D. 6 million. What percentage of SNPs occur in coding regions? A. 0.5%, B. 1%, C. 2%, or D. 5%? B. 1%. SNPs that do not affect gene function on phenotype are called functional variants, silent mutations, neutral variants, or dominant variants. Correct answer is C, neutral variants. When a SNP is in close physical proximity to a disease-associated polymorphism, it is said to be in A, B, C, or D. Correct answer is C, linkage disequilibrium. Copy number variations consist of differences in the number of single nucleotides, large DNA sequences, chromosomes, or D, cells. Correct answer is B, large DNA sequences. Roughly, what percentage of CNVs or copy number variations involve gene coding sequences? 10%, 25%, 50%, or 75%? Correct answer is C, 50%. Differences in phenotype among monozygotic twins are most likely due to A. Genetic variations, B. Epigenetic factors, C. Non-coding RNAs, or D. Structural mutations. Correct answer is B. Epigenetic factors. Euchromatin is characterized as being A, B, C, or D. Correct answer is B, dispersed and transcriptionally active. What structure is essential for chromosome segregation during mitosis? A. Telomeres, B. Introns, C. Kinetochores, or D. Promoters. Correct answer is C. Kinetochores. The telomeres of chromosomes serve to encode proteins, link chromatids together, allow repeated replication without damage, or enable transcriptional activity. Correct answer is C, allow repeated replication without damage. In chromatid organization, the P arm refers to A. Short arm, B. Long arm, C. Central region, or D. Promoter region. Correct answer is A. Short arm. What sequence helps initiate gene transcription? A. Telomere, B. Promoter, C. Enhancer, or D. Exon. Correct answer is B. Promoter. Which DNA regions are removed from pre-mRNA during processing? Exons, introns, promoters, or enhancers? Correct answer is B. Introns. Non-coding DNA elements that can influence gene regulation includes exons and codons, introns and promoters only, short repeated or transposon, or centromeres and telomeres. Correct answer is C. Short repeats, regulatory RNAs, and transposons. What is the role of epigenetic factors in differentiated cells? A, B, C, or D? Correct answer is B, to regulate lineage-specific gene expression. Which protein complex allows the genome to be compacted within a small nucleus? Ribosomes, chromatin organizing factors, histones, or DNA polymerases? Correct answer is C, histones. What structure resembles beads on a string in chromatin? A. Nucleosomes, B. Centromeres, C. Telomeres, or D. Introns? Correct answer is A. Nucleosomes. Chromatin structure affects gene expression by changing the DNA sequence, regulating the accessibility of transcription sites, replication, additional copies of DNA, or silencing genes. Correct answer is B, regulating the accessibility of transcription sites. Chromatin remodeling complexes primarily function to A, B, C, or D? Correct answer is B. Reposition nucleosome to expose gene regulatory elements. Histone acetylation generally results in chromatin condensation, increased transcription, gene silencing, or chromosome replication. Correct answer is B. Increased transcription. What enzyme removes acetyl groups from histones leading to chromatin condensation? Histone methyltransferase, DNA polymerase, HDAC, or topoisomerase? Correct answer is C. Histone deacetylase or HDAC. Phosphorylation of histones on specific residues can lead to A, B, C, or D. A. Either transcriptional activation or inactivation. DNA methylation in gene regulatory elements usually leads to gene activation, transcriptional silencing, increased mutation rates, or decreased DNA stability. Correct answer is B. Transcriptional silencing. Which enzymes add methyl groups to DNA resulting in gene silencing? Demethylases, DNA polymerases, methyltransferases, or kinases? Correct answer is C. Methyltransferases. HDAC inhibitors are being tested for the treatment of A. Diabetes, B. Cancer, C. Heart disease, or D. Alzheimer's disease. Correct answer is B. Cancer. Nucleosomes consist of DNA wrapped around A. RNA, B. Histone proteins, C. Lipids, or D. Carbohydrates. Correct answer is B. Histone proteins. What is the length of DNA wrapped around each nucleosome? 147 base pairs, 100 base pairs, 80 base pairs, or 50 base pairs? Correct answer is A. 147 base pairs. Which histone subunits make up a nucleosome octamer? A, B, C, or D? Correct answer is B. H2A, H2B, H3, and H4. The positive charge of histones helps them bind to A. Negatively charged DNA, B. Positively charged DNA, C. Cell membranes, or D. Neutral lipids. Correct answer is A. Negatively charged DNA. What is the primary function of microRNAs or miRNAs? Encode proteins, translation of target mRNAs, replicate DNA, or enhance DNA repair? Correct answer is B. Modulate the translation of target mRNAs. Micro-RNA post-transcriptional silencing of gene expression is observed in A. Only plants, B. Only animals, C. Only fungi, or D. All eukaryotes. Correct answer is D. All eukaryotes. What is the approximate length of mature microRNAs? 5 nucleotides, 22, 100, or D. 200 nucleotides? Correct answer is B. 22 nucleotides. How many microRNA genes are encoded in the human genome? A. 100, B. 500, C. 6,000, or D. 30,000 genes? Correct answer is C. 6,000 genes. What enzyme trims the pre-miRNA to form mature miRNA? A. Polymerase, B. Helicase, C. Dicer, or D. Exonuclease? Correct answer is C. Dicer. Mature microRNA associates with which complex to silence target mRNA? A. Polymerase complex, B. Replication fork, C. RNA-induced silencing complex, or D. Ribosome? Correct answer is C. RNA-induced silencing complex or risk. miRNA binding to mRNA can lead to which outcome? A, B, C, or D? Correct answer is B. mRNA cleavage or repression of translation. Small interfering RNAs, siRNAs, are used experimentally to increase DNA transcription, knock down specific gene functions, replicate RNA, or delete histones. Correct answer is B. Knock down specific gene functions. Long non-coding RNAs are generally longer than 22 nucleotides, 50 nucleotides, 200 nucleotides, or 1000 nucleotides. Correct answer is C. 200 nucleotides. Which of the following is the role of long non-coding RNAs? A, B, C, or D? Correct answer is C. Modulate gene expression by various mechanisms. Which long non-coding RNA plays a role in X chromosome inactivation? A, B, C, or D? Correct answer is C. Zist. Long non-coding RNAs can silence genes by destroying mRNA, binding to chromatin to restrict RNA polymerase access, initiating DNA replication, or D. Enhancing ribosome function. Correct answer is B. Binding to chromatin to restrict RNA polymerase access. siRNAs are commonly used to study gene function by enhancing gene expression, silencing specific mRNAs, editing DNA, or D. Transcribing RNA. Correct answer is B. Silencing specific mRNAs. Which protein complex is involved in the unwinding of double-stranded miRNAs? A. Ribosome, B. RISC, C. Dicer, or D. Polymerase? Correct answer is B. RISC. What type of immunity does the CRISPR or Cas system provide to bacteria? A. Active, B. Acquired, C. Passive, or D. Natural immunity? Correct answer is B. Acquired immunity. In gene editing, what is the function of the guide RNA or gRNA? Cuts DNA directly, transports DNA segments, binds and directs Cas9 to specific DNA sites, or repairs DNA strands? Correct answer is C. Binds and directs Cas9 to specific DNA sites. How many bases are in the guide RNA sequence used in gene editing? A. 15, B. 20, C. 25, D. 30. Correct answer is B. 20. What is the potential application of the CRISPR or Cas9 system? A, B, C, or D? Correct answer is C. To insert mutations for disease modeling. Which enzyme does Cas9 resembles in its function? A. Ligase, B. Nuclease, C. Polymerase, or D. Helicase? Correct answer is B. Nuclease. Cas9 uses which part of the gRNA to recognize specific DNA sequences? A. Constant region, B. Variable region, C. Coding region, or D. Silencing region? Correct answer is B. Variable region. CRISPR sequences in bacteria provide immunity against A. Viruses only, B. Phages and plasmids, C. RNA viruses, or D. Fungal infections? Correct answer is B. Phages and plasmids. The final question. What mechanism allows for the precise insertion of new genetic material? A. Non-homologous end joining, B. Base excision repair, C. Homologous recombination, or D. Mismatch repair? Please comment down your answers. And lastly, if you learned something new from this video, please don't forget to subscribe the channel. That would be much appreciated. So that's all from my side for now. You can continue your learning by watching our next video.